All right, good morning, everybody. Hello. Welcome back. For most of you, I assume. For those of you that are new to campus, welcome to genetics. I'm Dr. Ross. Hopefully everybody's in the right room. We have to do this, right? It's required. You're supposed to be somewhere else. Philosophy? No. We'll go a little bit deep in a little bit, but it won't get too philosophical. Okay, so everybody's in the right place. Got all the chairs filled. Excellent. So hopefully everybody's got Socrative, or at least a web browser. So you can go to Socrative.com. We'll be doing this later in the class with your tablets. So hopefully everybody's got their mobile device ready to go. This is a Discovery class. We do have Wyatt from the Discovery Hub here to give us help later if we need it. So everybody's got tech support. We'll talk more about tech support in future. And I'm going to do this sort of thing at the start of every class. So everything here is really done deliberately. I'm a really super objective person. I like to make decisions based on data. Hell, I'm a scientist. I close the blinds not because I don't want you to be distracted by the beautiful people walking by outside or the biology that's happening outside, but because of glare on the screen. So we're here. It's Biology 102. Today is going to be a lot of talking, but that's going to be by me, unfortunately. That's going to be kind of abnormal for this class. You'll see what I mean in a little bit. I did have this great video to show you. You should actually go watch this video. It's only 2 minutes long, and for some reason, the audio is not working today, so we adapt, just like biology. But this is a fabulous video. It's really brief, and it discusses why it's important for all of us to be practicing scientists and to practice critical thinking. And that's really what we're going to be doing in class this term. Yeah, it's genetics. By the way, who's taken 101? Biology majors, Biology 101? Okay. So for some of you, this is among the first upper division major classes you've taken. Things get different. Hopefully you think it's a good different. It's going to be less fact memorizing, more application, more understanding, more practicing, less multiple choice. So for some of you, this is going to be uncomfortable. For others of you, this is going to be a welcome relief. But Carl Sagan wants us to know exactly why it's important to practice those critical thinking skills, the analysis skills, and I do hope you'll watch this video when you can. You've all got tablets, so you can do it after class if you want. So we're not going to be doing multiple choice, or at least not much multiple choice. One of the most important things I want you to keep in mind the whole semester, especially for those of you that are biology majors, because I'm a biology faculty member, I'm most familiar with our major. Apologies to those of you that are chemistry majors and so forth. But this probably applies to Chem 1A and 1B as well as Biology 1A and 1B. Get comfortable with this idea. We're not talking about facts. There are some facts to be learned. The emphasis here is not on facts. The perspective that I hope you will all have, starting the term, but especially by the end of the term, is that we don't know the right answer. There might not even be a right answer. You might see some questions on exams where you think, is this a trick question? I can't figure this out. That's what real science is like. Sometimes there is no right answer. So we're going to be practicing science the way we experience it as professionals, just memorization. So Carl Sagan wants us to do critical thinking. Okay. So what exactly do I mean? What is my goal for all of you for the end of the term? How do you solve a problem? It's how to be presented with data. I'm sure I was there too, especially as an undergraduate. I read an exam question and I think, I know there's some distractor material in here. There's like a formula that they put in there that I don't really need to use. Has anybody experienced this? Right? They're telling me this information to distract me, to try to prevent me from getting the answer. Well, in reality, of course, when we do research, as biologists, as geneticists, we have to figure out what information we need to start with. We need to know what sort of an experiment to design, and so critical thinking is partly trying to figure out what information do you need. That's something we're going to emphasize in class. Using data to support a decision. So it's not just going to be regurgitation by you of facts. That's not what I want. what I'd rather have you be able to do by the end of the semester is to get data, say, I think that this is what we should do and this is why. And support that why with fact, to make a claim based on evidence. Say, I think we should have transgenic crops because this data suggests that it's good. And I'd also like you to be comfortable to have the opposite, to be able to reflect the opposite opinion as well. So I'd love to have everybody become a balanced arguer. I have this evidence that suggests that transgenic crops are good, speaking of genetics, and there's this conflicting evidence that suggests that they're not good. So let's have a discussion, let's debate, let's see which evidence outweighs the other. Let's make a decision and move forward. So in order to accomplish this, I've just listed a few different things that I think you should bring with you if you really want to be prepared to excel in this class with me as the instructor. The ability to think about what you've done, to self-evaluate and to error check. So not just to do something once and say, oh, that must be the answer, and move on. Think critically about what did you just do? Was it right? Was it appropriate? And most of all, please, please, please be creative. And everybody sits there thinking, what the hell? This is a science class, right? Creativity has a key role in all aspects of biology, especially genetics. The part where creativity comes into play, and I hope those of you that think you're more creatively minded will really shine here, is that when we observe something, there is not just one right answer. There are many ways to interpret data, and you have not just the right, and when you watch the Carl Sagan video, you'll understand what I mean by you basically have the obligation. You are obliged, as a good citizen of this world, to question everything. Please question me. I love questions. That shows me that you're learning. I prize question asking more than almost anything else in the classroom. By the way, does anybody have any questions at this point? It's been a lot of this by me. Any questions at all? Why did I sign up for 102?
What's that timer at the bottom? Are you recording?
Yes. So one of the benefits of being in a discovery classroom is that I am recording everything that I'm saying and everything I'm doing. And after class, so for example, I've got a laser pointer tool. I can direct your attention to certain parts of the screen. All of this gets uploaded to YouTube after class as a movie. So you get all the audio, and you get whatever I do on the surface of the tablet. So if I write on a slide, if I annotate a slide, all of that becomes a movie. It's on YouTube for you to use. And we'll talk more about this next class when we go into more mechanics of the class. This is a great resource. I hope that you will all join us in class every time. If you can't, watch the video. It won't be quite the same as being in class because we will do some things that I don't record, not because I don't want to, but because I can't. But this is an excellent resource. So on your syllabus, which hopefully everybody's seen at this point, there's a link to my YouTube channel. So you've got the URL. Go there, you can start watching videos. In fact, that's where you'll find that Carl Sagan video. There's a link to it on my YouTube channel. Check the syllabus, YouTube channel, Carl Sagan video, and everything that we produce this term is videos from our tablets. We can post there. Other questions? By the way, of all the things that I've done in my previous tablet classes, this one thing, lecture recording, was the most highly valued by all the students I've ever had in a discovery class. Because it's not just lectures I record, office hours sometimes I record. If the person that's sitting in the office with me is comfortable with that idea, it's just a recording audio, no video, right? You can't see me. Nobody really wants to see me in a video anyway. So it's just what's happening on the tablet and audio. But I love this because when a student comes and asks me a question, this always happens. A student comes into my office and asks me this really fabulous question, and I give the most beautiful explanation on the whiteboard, and then what happens? 5 minutes later, the next student comes in with the exact same question, and I say, oh, if you had only just been here about 3 minutes ago, you would have seen the perfect explanation of this. So now I record all of this. So I can post it on YouTube. All of you benefit from the question that one student asked. So if you're not comfortable with asking questions in class, but you share that question with somebody else, you can benefit from that resource. Okay. So there will be no syllabus anything today. It's missing from the outline. A little bit next class period, maybe. Today's all about setting atmosphere and getting to know each other. And so I'm going to start with some announcements. This is weird. I'm not usually going to do announcements in class. That's what Blackboard's for. I'll do a brief intro of myself. I'll talk a little bit about how the course has been designed, and then we'll talk a little bit about tablets, start to use the tablets. This week is about being comfortable with using the tablets and getting into a little bit of genetics content. Next week, we go full force into practicing genetics. But we've all got tablets especially, so there's no reason I have to stand up here telling you announcements. So announcements are always going to be posted on Blackboard. So do check Blackboard regularly for announcements. Because we haven't started yet, I get to do in-class announcements today. Come Friday with your tablet fully charged for a quiz on the syllabus. So read the syllabus, make sure you understand it. It's not going to be a difficult quiz, mind you. Exactly how many points did you have to get a B? No, nothing like that. Simple stuff. I want to tell you that there might be assigned seating in the future. Today you pick your own seat, maybe not in future. We'll see. There was a mistake on the syllabus. I already told you to go to my YouTube channel. This is the real URL for my YouTube channel. If you've got one of those QR code scanners, take a picture of the screen. It'll take you to that URL with that fancy little 3-dimensional QR code that's 2-dimensional. I'm not a physicist. Really important point. I pride myself, among many things that I do, I pride myself in putting the lecture slides on Blackboard the night before class. So everybody comes with the ability to pull up the exact PDF of the slides that I'm using and take notes on them. I hope you will. Taking notes on the slides is one of the really outstanding routes to success in this class because of the way I teach. This is a lot of text, but most of the slides we're going to see when we're actually talking about genetics are mostly diagrams and figures, not so much writing. So I'd like you to write your interpretations, your questions, your ideas, your thoughts, digitally if you want, or print them out and write pen or pencil and paper if you like in class. Note-taking is really critical. And as I said before, the videos I'll post to YouTube. Oh, now it gets personal. What's it like being me? So this is actually an MRI of my brain. How many pre-meds do we have in here? People that think they want to be doctors. Okay. Does anybody see, not just the pre-meds, I'm just getting a little census here. Does anybody see anything asymmetrical about my brain? Something look weird? Who sees it? Anybody want to be brave and point out part of my brain? Sir, here. Lays my brain.
Like this part right here in the left looks different.
That's right. My right temporal lobe's got something weird going on. And a decade ago, I had that thing taken out. Not a tumor, but something like it. So when I was a graduate student, I had brain surgery, and I'm just telling you this not only because it's an interesting icebreaker, and I don't want you to share your medical issues with me unless you feel comfortable, is that it turns out that the right temporal lobe, right behind your right eye, that part of the brain stores emotional memory and visual memory, and I'm using this as a cop-out because I'm really horrible with names and faces. So I do hope you'll forgive me. It's a big class, and I'm going to try to learn everybody's name and everybody's face. That's my goal. And I'm going to do something with you with the tablets as homework for next class that's going to help me out a lot with this. Other than that, I'm perfectly normal, as you can tell. You'll figure it out. So speaking of perfectly normal, again, by way of introduction, in talking about critical analysis, I declare that I'm an objective evidence-based decision maker, and this is evidence of that. Here's a plot that I made last year. I've taken the axis labels off. What do you think this is a plot of? You can kind of see part of the legend over here on the right. 2012, the diamonds. 2013, the squares. Poly, that line is a polynomial regression, a best fit line through the red data series. What do you think this is a plot of? Okay, 30 seconds. Talk to your neighbor. Figure out what you think. I want a proposal. What do you think this plot is of? Go. Talk. Make noise. We'll come right back. Okay, so here's a hint. It has nothing to do with genetics. Sorry. Okay, so what do you think this is? I plotted this. I collected some data. I made a graph. Trying to do some critical analysis. I want to know the answer to something. What are the axes? What's on the x-axis, probably? 0, 60, 120, 180, 240. What units? Make up units, yeah. Could be time. Okay, and then when you watch the video, you're going to watch me write the word time on the slide. It's magical. What about the y-axis? No, I might actually predict it was the opposite, but we can go with number of students. So what does that suggest about, so that maybe this is biology 102 attendance. I don't know if that's what you're getting at. My handwriting is horrible. So we've got time, number of students. So let's see, 180 minutes. So over 3 hours of class, my attendance goes up. That's how I interpret this. So I start out at 0 minutes, I start lecturing to 40 students, and then slowly people get attracted by the sound of my voice. And holy crap, by the end, I've got the whole Science 2 building in here listening to what I'm spouting off about. In reality, this was me cooking Thanksgiving dinner. And I wanted to know exactly, I had a potato dish that needed to bake exactly 45 minutes, and I wanted to know, when am I going to put the potato dish in the oven to cook with the turkey as the turkey's finishing cooking, so that they're done at the same time. So yeah, I measured the internal temperature of the breast in degrees Fahrenheit over time, and man, look at that data. That best fit line, 2013 was a great year, the red squares. I could predict to the minute exactly what time that turkey breast was going to hit the temperature that I wanted to pull the turkey out of the oven. And so you better believe, right about there, the potato dish went in. So, by way of introduction of me, this is the type of person I am. I'm really anal retentive, I'm really objective, I like to make decisions based on data. That's, I think, why I'm a successful scientist. I'm not trying to get you to this level, please. You can if you want to, it's fun. But this is what I'm trying to model for you as the instructor of this class. Collecting data, making decisions based on data. Now, a little bit more about me. I was an undergraduate at University of Oregon. I was a duck. I got my degree in biochemistry working on what do you think? Yeast. You don't get any degree just working on beer. That leads to not a degree, usually. In fact, I didn't really develop a taste for beer until I was a postdoctoral scientist. It was all hard alcohol until then. So after I graduated, in moderation, be responsible. So after I got my bachelor's degree, I moved to Seattle, where I worked for a couple of years doing 9 to 5 in the lab research work at the Fred Hutchinson Cancer Research Center, which is a really cool place to work, and Seattle's a really cool place to live, on mouse noses. Specifically, how proteins allow mice and humans and other organisms to detect smells. So I was studying the senses, essentially. Right, so I've worked on yeast, I've worked on mouse noses. I got my doctoral degree, my PhD, also in Seattle, working on this particular species of fish, the three-spined stickleback fish, which is a really cool fish for a lot of reasons, both genetically and evolutionarily. For those of you that are going to be taking Biology 105 evolution someday, you might hear more about sticklebacks then. We have a local population. They live in the San Joaquin River. You can go to Friant right below the dam, and you can actually go wander around and see sticklebacks. And then after I got my PhD, I spent 4 more years in school, kind of, doing postdoctoral research on this fabulous nematode, which is not to scale. It's actually about 1 millimeter long. And the reason I'm telling you all of this is because you're going to see a lot of examples that I draw on in class from these specific systems. Olfaction, proteins that let us detect smells, yeasts, sticklebacks, and my favorite species, Caenorhabditis briggsii. Pronunciation beyond exams. So I'm here, part of my job is teaching. Right? You may or may not know that faculty in the CSU system, our jobs consist of about 40% teaching, 40% research, and 20% serving on committees and doing other administrative work of the university. And this is what I do in that 40% of the time that's research. I have a lab across the street on the 3rd floor of the Science 1 building. These are all of the students that I had working with me, mostly for credit, some very few for money. But last spring, this was my group of students that worked with me. And we all work on this worm, Caenorhabditis briggsii, and that's why a lot of what you're going to hear about this term is related to the genetics of this particular species. Okay. So briefly, the question that I'm interested in terms of genetics, personally, doing research across the street in the lab, is how you tell what's a species from what's not a species. How do we know who are members of the same species and who are members of 2 different species? Does anybody have any ideas about how we do that? If I open the window, you'd look out the window and you'd see all sorts of different trees. They've all got leaves, mostly. Are they all members of the same species? So leaves isn't enough. Like, plants have leaves. Most plants have leaves. So how do we know that these dogs are all members of the same species and these Darwin's finches on the right are all separate species? How does the biologist know who's a species and who's not? Oh, genetics. DNA, yes, absolutely. So we could extract DNA from everybody. If we wanted to know if we were all members of the same species, we could take DNA samples of everybody. We could come up with some definition of whether or not we're all members of the same species or not. There's something more practical and more fun we can do with these organisms to tell if they're the same species or not.
Observation.
So what would you observe? Observation.
Hair color, teeth, etc.
Okay.
Structure.
So visual. So especially in Darwin's finches,
Right.
the shape and morphology of the beak suggested to Darwin that they were different species. Right. We can look for different coloration, but then again, we've got an issue. Go ahead.
If they can successfully reproduce?
The biological definition of species. So we've talked about a genetic definition of species. We could get DNA information and we could just make an arbitrary decision who's members of one species and who is a member of a different species. We can look at morphology, or we can mate 2 individuals together and find out if they have living, viable, and fertile offspring. That's the biological species concept, this one down at the bottom. If you mate 2 individuals together and they can have kids, and their kids can have kids, members of the same species. Is that true for dogs? There's a lot of diversity here. Look at all those different breeds of dogs. What happens if you made a Labrador with a poodle? You make a Labradoodle. So their offspring is alive. Are they fertile? Some are. You don't have to know that. This is not in the quiz. So this defines the Labrador and the Poodle as members of the same species because they have viable, living, and fertile offspring. So Labrador, a dog. Poodle, a dog. Same species. On the other hand, here are two canids that look, to me, on the top here, they look more similar to each other than a Labrador and a Poodle. Different colors, different sizes, different hair textures, and so forth. Are they the same or different species? Coyote and a German Shepherd. What's the question you should ask? If
Is a child for a time?
is the child, so the child is alive, that's the picture down here, their offspring. So yeah, you want to know, are they fertile? This is good critical thinking. If you want to know, are these two species, mate them together, look at their offspring, and ask that critical question. It turns out that most, and I did not make this up, I know Labradoodles most people know. This F1 individual, first generation hybrid, it's called a Kai dog. I didn't make it up, that's just what they're called, half coyote, half dog. They're sterile. Okay, so despite the fact that a German Shepherd and a coyote physically look pretty similar to each other, size, some mannerisms, they are members of different species. So that's the question that I'm interested in studying in my lab with my worms. So not with horses and donkeys, but you're probably most familiar with this case. You mate horse with a donkey, and you make a mule, and the mule is sterile. And we know, now we're getting into genetics, we know why the mule is sterile. It's because the horse has 32 pairs of chromosomes, every cell. Donkeys have 31 pairs. So my question to you, just think about, maybe we'll talk about this at the start of next class. If one parent has 32 pairs of chromosomes and the other parent has 31, how many pairs of chromosomes does their offspring have? Don't answer, just think about it for next class. Thought assignment. We'll pick this up when we start again. So it turns out that the answer to this question is the reason why the mule is sterile. And in my lab across the street, come visit sometime, I'll show you the lab. Instead of using horses and donkeys because they're big and smelly and expensive, we use these microscopic nematodes. The adults one pictured here is about actually a millimeter in size. And so you'll often see most of those students that were in the picture sitting at desks, hunched over microscopes, looking at worms and mating them. So what we do is we take worms from all over the world, members of this species, and we mate them together, and we find out which of our worm populations are essentially the horse and the donkey of the worm world. Which combinations of mating lead to viable fertile offspring, that defines members of one species, and which combinations of parents from different parts of the world produce dead offspring or what? Dead or sterile offspring. And we do a number of other things too. The last little bit about me. Again, trying to get to know you, trying to get you to know me. This is my dad. This is my nuclear family. My two sisters on the edges, my dad, my mom next to him in the yellow shirt, and me. Okay. My dad is my motivation for being a teacher, and I just want to spend a couple minutes on why I think I'm here. Okay. My colleagues hired me to be an instructor in the biology department because I love to teach. I got that. This is kind of blasphemous for a geneticist to say. I inherited that from my dad. The love of teaching, I inherited from my dad. My father has been a community college biology instructor since 1968, which means, yeah, he's pretty old. I'm not that old, but he's pretty old. And he retired about a decade and a half ago and still teaches. Kind of like Dr. Schreiber, if you know Dr. Schreiber, right? Lots, when I talk to Dr. Schreiber, I have to restrain myself because I want to say, you know what? That reminds me a lot of my dad, but I don't really want to go there because Dr. Shriver, I just don't want to make the age difference between he and I that apparent. But in a lot of ways, they're very similar. They love teaching. They can't let go. So Dr. Shriver is officially retired, but he's still here teaching. So he's teaching the other section of genetics starting in half an hour. And so you'll see him if you stick around. Okay, so in a nutshell, I love to teach. I love seeing the spark in the eye when a student really gets that concept that I'm trying to get across. And more importantly, I feel like it's my responsibility to help people. I love helping people. And that's why I think this is a good leverage of my background in genetics and biology and the fact that I get to teach lots of you. And I get to try to help you. So, blah, blah, blah. Totally guilty of conflicting evidence. I've been talking now for half an hour. My role as your instructor is absolutely not to stand up here every class and yak. I do not want to lecture. I don't want to profess, even though I'm a professor. Talking is boring. You don't learn by me talking. I certainly don't want to put anybody down. I'm not here to intimidate. I'm not here to make things exclusive. This is definitely not a weed out class. I hope everybody gets an A, and you'll see that when you see the grading on the syllabus. At the same time, I do not give points. You earn points. And hopefully this is not uncomfortable for you, but I don't feel like I teach, right? I'm not here to give you information. I'm here to help you learn your own information. This is more me, I think, kind of suave and debonair. So I don't want to be the professor. I want to model, not literally. Well, maybe someday. I don't know. If I got some hair putty or something. Except I bike to work, so it'd get all messed up, but I guess that's the look these days. I'm here because I've got lots of experience in biology, so I can hopefully give every one of you, to a certain extent individually, background on why this material should matter to you. Right? To make everybody understand why it's important to learn this material. To help you become better self-learners. I said I value questions, so I'm going to ask a lot of questions of you. And as I said before, I really appreciate the feedback you'll give me. If you ask questions, that's fabulous. I would rather have an entire 50-minute class period where you ask me questions and I give you answers, or I ask questions back to you, than me to prepare 50 minutes worth of me talking. That's definitely not what I want to do. Okay. Why is it important for you to become lifelong learners? You're probably thinking right now, I don't even have to ask you to raise your hands. I'm paying how much? And he's telling me that he wants us to teach ourselves? So there's a lot of evidence behind this decision that I've made on how to structure this class. The jobs that are growing most in terms of numbers of available jobs did not exist a decade ago. Think mostly IT, but also in biology. So information technology, networking, databases, computer programming, but also in the life sciences. So how am I going to help you? Presumably everybody wants to get a job when they leave. Maybe that's why you're here. You've heard that it helps you get a better paying job when you get a bachelor's degree, or it helps you take the next step in education. How am I going to help you succeed if I'm trying to prepare you for a job that doesn't actually exist right now? I don't know what to teach you. What possible content could I give you that prepares you for a job that doesn't exist yet? Because by the time you're 38, if you're not already there, the average person will have held that many jobs. So it's more important for your long-term success for you to learn how to adapt and to teach yourself new skills than it is for you to remember the 20 amino acids. Honestly. Right? It's not about content, it's about skills. Evolve or die, folks. Be adaptable, teach yourself new skills, and come to class prepared to practice those skills. So, in terms of what we're going to do in class every day, I'm not modeling right now what we would do in class every day. I'm just explaining it so you understand. Hopefully that makes everybody more comfortable about what we're going to do. Blended learning means I'm asking you, and you'll see this on the syllabus, to come to class having read the material already. So you read the material from the textbook before you come to class the day we're supposed to cover that content. That means that in class, I don't have to give you the content. You've already accessed the content. We spend in-class time, you and I, practicing, playing with the data, with how to solve problems, right? Doing the actual process of science in a lecture classroom as much as we can. And the tablets is going to help out with that a lot. We're going to be able to do a lot more with computers in our hands than we can with pencil and paper in our hands. And that's why I'm teaching this class as a tablet class. I hope you love this. You might not be happy with graphs. I've already shown you one, turkey temperature. I'm expecting this to show up on my course evaluations, by the way. Sometimes weird stuff shows up on my end-of-term evaluations, like all I learned about in this class was turkey temperature and how to... One of you is going to do it, I bet. So how does me lecturing to you, if that's the way I want to proceed, help you learn? These fabulous electrical engineers, sorry, which you can't see because I covered up the citation there at the bottom, Poe, Swenson, and Picard in 2012 developed this fabulous wristband that you can wear for 24, 48, 72 hours. And what this wristband tracks is, as it says down there, emotion, cognition, attention. Basically, how much is your brain being stimulated, activated at different points during the day. So they could track one person across day 1, day 2, day 3, day 4, day 5, and look at y-axis, brain activity. So you've got time on the x-axis, we've got how stimulated is this person. And you probably can't see the labels that are underneath the x-axis. So let's pick day 4. We've got lab here. So they were in a class that was a lab class. Any brain activity? A little bit at the start, then none, then a little bit at the end. Then they watch TV. Brain activity? Flatline. Studying? Maybe a little bit. Then they go to sleep. I'm not a neurobiologist. I suspect that's maybe REM or something like that. Not the band. Rapid eye movement. Dreaming. You with me? Okay, so sleep. Flatline. Then what? What is this yellow? What does it say for those of you that are in front that can read this? Class. So look at all of the classes that this student took. What does it look like? What other part of the graph does class look like? TV time. Passively sitting, watching, not participating. Like what you're doing right now. So I don't imagine you're going to remember a lot of this if I test it, which I'm not. If I tested this on the final, you probably wouldn't remember a lot of it because you're not actively involved in doing it. And that's why not only are we doing flipped classroom, not only are we doing flipped classroom, you prepare yourself in advance so that we can spend the valuable time that you're paying for in class with me, doing things with me, but we're also doing active learning. You're being a participant in the process, and that's what I really hope everybody will come back next class prepared. Well, next class we'll talk about the syllabus some. Definitely next Monday, full force, active learning, doing things in class, small groups, activities. There will be some lecturing interspersed, but I'm hoping that it's not just yakking at you. Okay. I'm also going to try to integrate for you to make things relevant so that hopefully you'll remember most of the information we cover, at least some of it. Give you authentic experiences in genetics. What's it like to be a real geneticist? A real geneticist does not sit in a classroom and listen to somebody talk. A real geneticist is doing experiments, analyzing data, and I hope this doesn't freak anybody out. I'm going to try to do group exams, or at least collaborative exams. Don't hold me to this yet. I'm still trying to figure out how to do this. My goal is to have open book, open note, open internet exams. No cheating. Because there's no way to cheat. Scientists don't call that cheating. I don't sit in my office alone. Well, I do. I have a wife and kids, by the way, but I do sit in my office alone and think about genetics. But practicing geneticists collaborate with each other. They talk to each other. I don't solve any problems on my own. I reach out to my colleagues across the nation or across the world and find experts to help me solve problems. Why should I keep doing what we've done for decades in higher ed and testing each of you individually on your own individual knowledge? Maybe it works, but for some of you it doesn't. So why not try collaborative exams? Okay. And mobile technology is going to help us do all of this. So the main ways that we'll use the technology are, A, it gives us access to the internet. We all have the internet, hopefully, we'll see. Hopefully we all have the internet access in class. So if I asked you to go find me some information about genetics in a database and I showed you how to use that database and all of that, you could get on a web browser and you could go do that in class, in a small group if we wanted to. Okay. The other thing I really love about teaching tablet classes is I get to use the tablets for you to give me feedback in a really comfortable way. Some of you are probably like me, introvert. There are two definitions of introverts, right? I don't look like an introvert right now because I'm comfortable standing up here and talking at you for an hour. But what an introvert means, I think, to a psychologist is somebody who doesn't like to give an answer right away. We get a question, we like to think about it for a while and make sure that when we give an answer, it's really carefully worded so that we don't make ourselves look stupid. So I want to give introverts and extroverts the chance to think about answers to questions before they actually ask them out loud, so we can use the tablets to do that. And next time we'll talk more about this. I love to use Twitter, so everybody should be hopefully on Twitter, or at least have an account. It's free. You can tweet me questions during class if you don't want to raise your hand. 140 characters. /2 of 3, 3 of 3, whatever you need. Totally fine. So we have a number of technologies that we'll use, including Twitter, for you to send me questions, and for you also to get feedback about how you are doing in class. So this is just a short list of some things that you can expect us to do with tablets this term in the class. The normal stuff, you'll read PDFs like the lecture slides, you can take notes on the lecture slides, watch videos, but there are a lot of other things we're going to do too. Although tablets are required, this bottom point is really important. I want to make sure it's really clear that tablets is not a gimmick, and I am not always going to use a tablet. I'm going to use it when the situation warrants using a tablet. There are some times when we're going to do things that we won't use our tablets for. But you should still always bring it fully charged, even though we have these lovely, somewhat dysfunctional electrical outlets in this classroom. Don't expect you can charge your tablet in class. So come prepared with it charged, and we'll use them. So in the last 5 minutes, I've got one tablet exercise, but just before that, I want to share a little bit of homework with you. 2 easy assignments, hopefully easy, just to make sure that your tablet is working properly. So for participation points for today, there are 2 assignments, the 2 bullet points there. I've set up a Google Classroom, and for those of you that haven't used Google Classroom before, it's really straightforward. Go to the syllabus for the class. The URL for signing up for our Google Classroom, it's very much like Blackboard, but very different, is on the syllabus. So go register for yourself in our Google class, and there are 2 assignments already waiting there for you. The first one helps me with my brain issue, which is I'd love for everybody, please, to take a selfie with your tablet and send it to me. This helps me because I've got your face and I've got your name. So I can flashcard, basically. I can start trying to memorize people's names, associate them with the faces. Hopefully that makes us feel more comfortable with each other, asking questions and interacting in class. The second one is a PDF annotation exercise. I uploaded a essentially blank PDF file that has a few questions written on it. So that exercise is going to ask you to download the PDF, draw or write your answers to the questions, and then send me that file through the Google Classroom. So by next class, sign up for Google Classroom, do the selfie, do the PDF annotation exercise, and then lastly, find yourself a QR code scanner, free. Don't pay for one, they're free ones. I use Crafter, but I use an iPad too. So if you have another operating system, you might have to find a different app. Any QR code scanner would be fine. 5 minutes left. Time for one question. Plenty of time for questions next class.
So you want to send us off with your email?
Not through email. So the way the no. You can if you want to, but the way the Google Classroom works is you log on through the web to the Google Classroom, you poke on the assignment, you can attach a file. And it uploads, it's all through Google Drive. So everybody has a mail.fresnostate.edu account. You all have Google Drive accounts. And so when you take a picture with your tablet, it'll save it on your tablet. You can attach that picture to the Google Classroom homework assignment. And it sends it directly to me, not through email. Good question. One more question. Saw one more. No? It was the same question. It's magical. Okay. So, last thing. With a word of caution. So, I love this app, Socrative, that hopefully everybody has installed. If you don't have the app, you can use a web browser to go to b.socrative.com. It's exactly the same thing. You don't have to have the app. It'll ask you to join a classroom. 240820 is the number of our classroom. Technically, Socrative only allows 50 students, which it says down here in the bottom, to enter the classroom at a time. So we're going to test science. We're going to do an experiment and see if we can crash the system. There should be 75 of us in here. So there's a 3-question poll here. Just a second. I have to start it first. You can log in, but it won't show up yet. Okay, so poll is running. So if you log in, you should be able to answer these 3 questions. And when you're done, please log out in case somebody else isn't able to join the class because it's full. This is just one type of exercise we'll do regularly in class, not to do written response necessarily. To those of you that use clickers, this should look a lot like clicker technology, fancy clickers. All right. And if you don't know your student ID number, just put your name in instead. I prefer student ID numbers because sometimes people use nicknames that don't correspond with what I've got in the class roster. And if anybody has an issue, this is the point where we can enlist Wyatt from the Discovery Hub to help me troubleshoot any technical issues. So if you have any issues, raise your hand. I'll come around. And when you're done and you've logged out, please feel free to take off, and we'll be back in class on Friday.